So we are beginning with our next session. Our current speaker for this session is Dr. Umesh Kumar. He is currently working as associate professor in School of Business, IMS Ghaziabad University. He completed his bachelor's in life sciences from NREC College, his master's in MSc Biotechnology from Modi College, and PhD in Biomedical Sciences from Dr. B.R. Ambedkar Center for Biomedical Research, University of Delhi. He has done his doctoral thesis entitled Epigenetic Regulation in Forecast Carcinogenesis. He is currently having a DST-SERB project on genomic target-based chemical ligand profiling using natural products for identification of spot drugs for rheumatoid arthritis therapy. He has been honored with prestigious awards like Scientist of the Year 2020, ESDA Green Leadership Award in 2020, Young Scientist Award in Basic Science category, Senior Scientist Award in 2018. Currently, he is a member of various associations like Associate Member of American Association for Cancer Research, USA, Member of Indian Association for Cancer Research, Founder and President of International Interdisciplinary Researchers Federation. We are honored to have you, sir. The stage is all yours.
Thank you, Nikita. Let me share my screen first. Uh, yeah. Let me know if it is visible. Am I audible to everyone?
Yes, sir, you are.
My slides are visible.
Yes, sir.
Okay. Thank you, Nikita. So, very good afternoon to everyone. I'm really thankful to the organizers of this ICIBLS 2020, particularly Dr. Smita Das, Madam, and Professor Jagopal Sharma, Head of the Department. So, as I have been introduced by Nikita, so currently I'm working in IMS as Associate Professor in the School of Biosciences. Along with that, I'm working on some natural products-based compounds in IGIB, particularly in the field of cancer and rheumatoid arthritis. So I shall be discussing today epigenetic therapy in breast carcinogenesis that will be based on the natural products compounds, just because these synthetic compounds are a bit toxic, and those could actually interfere in different prognosis of the diseases also, right? So I'll be sharing my slides one by one. Let me do sharing just, yeah. So as you know that cancer is a significant global health concerns in non-communicable diseases. And 2018, approx 18.1 million new cancer cases were reported with this cancer, of different sites, alongside 9.6 million total mortalities have been reported. So that's a huge number that is actually a big concern, and it is assumed that by 2040 it will be actually doubled, particularly in low and middle income countries. So that is a big concern. As far as breast cancer is concerned, so it is one of the most commonly diagnosed cancer of the woman, that is actually a leading cause of the cancer death among those women, according for an estimate of around 6.24 lakhs death every year. So it is a big concern for as far as the breast cancer is concerned in the India. This is becoming a number one as far as the incidence and mortality is concerned, leaving behind the cervix cancer. And ultimately it is actually based on those different subtypes. Triple negative breast cancer is certainly rising nowadays, because that is actually showing very poor prognosis, alongside that neoadjuvant therapies, radiation therapies, and chemotherapies. So it is a matter of concerns for India also. So, what is actually epigenetics? This is a kind of heritable molecular mechanism that is actually controlled by those external factors that actually regulate the gene expressions without any alteration in the DNA sequence, actual DNA sequence. So this progression of breast cancer actually involve the accretion of aberrant changes, both at the genetic level and epigenetic levels, that is actually leading the tumorogenesis. So DNA methylation and this histone modifications actually form a epigenetic code that actually lead to the epigenetic silencing. And this DNA methylation is a critical enzyme derived chemical modification, that is addition of this CH3 group onto that CH3 group onto that cytosine or adenine of this actual DNA sequence. And then it is ultimately inhibit that gene expressions by the involvement of different actually enzymes like DNMT1, DNMT3A, and DNMT3B. So this is the case of those normal cells and cancer cells. Normal cells particularly are having actually those CPG rich regions at the promoter regions, and in the transcription start sites, and repeated sequences are not generally methylated. So this CPG island generally actually shows hypermethylation that actually cause the problem, that actually reverse the transcriptions, and that actually block the transcriptions. So ultimately it will lead to the silencing of those tumor suppressor genes. So this actually gives the idea about those kind of therapies which could actually alter this problem, because these are reversible processes. It can be reversed with some synthetic molecules or some natural compounds or dietary fibers, or some other pharmaco products. So it can be reversed, so that the transcription can initiate. So there are certain synthetic inhibitor like 5-azacitidine that actually is a DNA methylation inhibitor, but because this is a kind of toxic, so that actually gives a opportunity to deal with some natural compounds like curcumin, gensten, and silymarin. So these can actually alter these processes of DNA methylation and some histone modifications. So why epigenetic therapy? Just because uh, uh, these genetic alterations basically change the phenotype of the gene. They change the gene expression due to the mutations, recombinations, and transpositions in DNA sequences, and that actually causes the irreversible change. But in epigenetics it can actually be reversed by some epigenetic therapy, and normal gene expression can be achieved. So this cytosine methylation inactivate this condensed chromatin state by these processes of this DNA methylation. And this epigenetic silencing is actually involving a lot of enzymes, histone deacetylase, histone methyltransferase, HDAC inhibitors. So ultimately we can target these kind of inhibitors, which actually being inhibited by some natural compounds, and that can be very helpful to regain those gene expression of those tumor suppressor genes which has been silenced. So this is a kind of early event in this multi-step carcinogenesis, generally present in pre-malignant lesions. So that actually gives opportunity to to develop some biomarker on the basis of these tumor suppressor genes, which are getting silent by this hypermethylation of CpG island rich regions. So this is developing new strategies for managing the disease, that will be helpful to for the normal regulation of that tumor suppressor genes. So for that we have actually studied some of specific tumor suppressor genes. So among them, one of that tumor suppressor gene, that is GST-PI. So that is actually a gene which is actually hypermethylated in breast cancer, particularly in those breast cancer which are showing some estrogen receptor and progesterone receptor positivity, and that actually GSTs are basically the family of some isoenzymes that is actually interfere that is actually implicating in the carcinogen detoxifications. So it is actually conjugating with some xenobiotic, with some carcinogens, with some chemotherapeutic agents. Those are harmful. So it is actually reducing those the toxicity of that by conjugation of this glutathione with those xenobiotics. So certainly actually really actually doing that less toxicity of those xenobiotics, or eradicating those xenobiotics from the cell actually. So this we are studying, we have studied already this glutathione as transferase gene, GST-PI1 gene, because that gene is hypermethylated in 30%, 31% of primary tumor tissues. So it is of of very concerned gene in the cell cycle regulation also. So this gene, GSTP1 gene, is located on the chromosome 11, and this is carrying those CpG island rich promoter regions, which are normally methylated, but when it get actually hypermethylated in those healthy tissues, that actually causing the problem. And then this in the presence of that DNMT1, it actually becomes hypermethylated in that region. And certainly that these is transcription of that inhibits basically. So this is the process to become that a silencing of that particular gene, that is called epigenetic silencing. That certainly involves some histone modifications also, but this is a kind of method which can be actually reversed. So we have focused that reversal of this particular gene, and we have already some literature that is suggesting 31% of hypermethylation of GSTP1 in in primary tumors, while in some 44% of cell lines which are moreover ER/PR positive basically. So triple negative breast tumors are expressing this particular GSTP1 gene, but the that ER positive, PR positive, that means luminal A and B actually showing down regulation of this GSTP1 genes. So we wanted to see this GSTP1 genes, we wanted to reverse this hypermethylation by some natural compounds, and we use some polyphenols, and among them we have tried several natural compounds like curcumin, ECGC, genistein, isothiocyanates. So because these can alter, these are already reported that there are certain natural compounds which can actually reverse this process of DNA methylation in, in combination of other therapies also. Just because these are non-toxic molecules in certain concentrations, so it can be used to reverse this process. So this curcumin I have tried, and I have seen that this actually blocks the DNA methyl transferase ones to inhibit that DNA methylations. So as you know that curcumin is a yellow spice that is very bioactive component of perennial herb Curcuma longa. The problem with the curcumin is the absorption into the cells, but we have tried with uh, some vitamins like folate. So it shows that actually reversal. I will show you in the coming slides. But see the mechanism that the curcumin is a potent DNA hypomethylating agents that actually covalently blocks the catalytic thiolate of this DNA methyl transferase 1s to exert its inhibitory effect on DNA methylation. So ultimately, it is blocking that process through which that methyl group was attaching onto the cytosine base. So, I mean, the curcumin is having, three types of structure. So, curcumin 1 we have used, so that is actually exists predominantly in solution as the enol form, and that actual serves as a receptor to covalently block the catalytic thiol group in DNMT1. So this is how we are blocking this process by repressing DNMT1, by the curcumin, but in less concentration. I'll show you the which concentration. So this was the methodology we have used. We have treated those cell lines MCF7, which is ER positive, PR positive cells, and treated with the different concentration in the dose dependent manner and time dependent manner. So we have taken those MCF7 cells which has been treated with the different concentrations in 1, 3, 5, 10, 20, 30 micromolar concentrations for the 24 hours, 48 hours, 72 hours, and then we have checked the percentage viability through MTT assay. And then we have isolated those genomic DNA from these treated cells, and then we have modified the DNA. Bisulfite modification was done uh, just to check those methylation specific PCR for that particular gene. We have used the methylation specific primers and methylation specific primers to check the methylation of this gene. And then we have isolated simultaneously the protein whole cell lysate from those treated cells, and then we have done the western blot analysis to check the gene expression of GSTP1 genes. So then MTT assay has been done by seeding those 5,000 cells, treated cells, and we have seen that at 20 micromolar that IC50 value was calculated. And then because we have treated these MCF7 cells with the different concentrations of curcumin, 1 micromolar, 3 micromolar, 10 micromolar, 20 micromolar, and 30 micromolar. So what we have seen in the morphological changes up to that 10 micromolar from treated cells, we have not found those observable visible any visible change. But when the concentration was more, like 20 micromolar and 30 micromolars, so growth inhibition was noted, and certain cell death was reported. So this is how we checked the morphological changes, and then we have done those DNA defragmentation fragmentation assay by giving that 100 micromolar concentration of curcumin and then loading onto the 1% agarose gel. So what we have reported, actually, this is in case of GSTP1. So we have used those methylation specific primers to amplify those methylated band, which is actually in treated cells, MCF7 treated cells. So what we have seen in 10 micromolar concentrations, the methylation specific band was not observed. That is showing that that methylation has been reversed at 72 hours So it is, we have used those MCF7 cells untreated as, positive cells, and MDMB231, that is ERPR negative. So that those cells were used for the, negative control. So we have seen at 10 micromolar concentrations, we have reversed the, methylations. So simultaneously, we have observed at, at 10 micromolars, that, unmethylation specific band was observed. That means that, after the reversal of this, methylations, we have observed that band, unmethylation specific band, and that shows that, methylation, DNA methylation was actually, reversed. So this is how we have, reported that, reversal of GSTP1. Then it was confirmed by the, expression of the GSTP1 protein in 10 micromolar concentration at 72 hours. So the normal expression was reported at 10 micromolar, while at 20 microliter and 30 microliter, it gradually actually disappearing. So that shows that, that 10 micromolar concentration is the optimum concentration to reverse this process, DNA methylation process of, GSTP1. So by this, actually, I wanted to conclude that these kind of, polyphenols can act as, biphasic molecules. uh, at the optimum concentration, they can actually, give, the reversal of hypermethylation of that particular gene which we are targeting for that reversal. So this GSTP1 promoter regions actually showing that DNA hypermethylation, which is being reversed at, by the, 10 micromolar concentration of that curcumin, that actually causes the complete reversal of GSTP promoter hypermethylation. And with the, Western blot results, we can actually say that that actually leads to the re-expression of GSTP1. So simultaneously, we are checking with some more tumor suppressor genes like P16, HIC1, BRCA1, CDH1. So these are those specific tumor suppressor genes which are actually playing role in different, type of tumorigenesis in breast cancer. So they are actually involved, they are getting epigenetically silenced. So we are actually checking that if reversal of hypermethylation in those cell, genes can also be observed. So this require a lot of, set of natural plant-based compounds that could actually work in combination also, uh, uh, along with the neoadjuvant therapies. So it can be said that, these natural products can play a big role in epigenetic therapy of breast carcinogenesis. So this is just to show you that, this, this can be possible. uh, from this work we have, published this uh, in tumor biology. So and, this is a kind of, therapy through which we can reverse these processes which is causing epigenetic silencing. So with this, I want to thank all the organizers and, all the Asmita madam and Jagopal sir. So thank you so much. If any questions, I'll be happy to take.
Yeah, sir, we have a question. Are any in vivo studies being planned to study the effect of curcumin on methylation?
Right. So just can you repeat the question again?
Yeah, yeah, sure. Are any in vivo studies being planned to study the effect of curcumin on methylation?
No, it has been actually, under the trial, because as previous speaker told that, there are some nano encapsulations being done with these curcumin and other natural compounds, because this is, a kind of, compound curcumin that is not actually absorbed directly by the cells. So it has to be targeted, it has to be encapsulated through some nanoparticles or such kind of tools.
Okay, thank you so much. It
Thank-
was nice having you.
yeah, thank you so much, Nikita.
Now, we will be continuing our session after lunch break. Participants should join back for the upcoming interactive open group discussion and also in the concluding ceremony. We will be announcing the names of the winner of the award. So, everyone join us in upcoming session.